translation of basic biology leads to the improvement of human performance and health. Our basic knowledge on 1986 to 1977 on cardiac myosin predominance, left ventricle elasticity, metabolic performance,
היי, מחת.
לא, זה בסדר. לא, אין צורך, זה בסדר. אתם שומעים אותי כעת? Okay. Okay, so I'll start from the beginning. Translation basic biology leads to the improvement of human performance and health. Our basic knowledge on 1986 to 1977 on cardiac myosin predominance, left ventricle elasticity, metabolic performance, and acclimation mediated cross tolerance was translated on 1998 to a clinical study demonstrating that acclimatization to harsh desert environment can employed to human benefits. I'd like to thank, we are going to present this study, and I'd like to thank the organizers for inviting us to present this lecture. The start of my long adventure on acclimatization and heat acclimatization started when I heard a department seminar by a guest from Australia who was involved by the discovery of the cardiac isosomes, myosins. And I was attracted by one sentence of his expression that decreased thyroxine level enhances myosin metabolic energy efficiency. Because of my zoological background, I knew that desert inhabitants show apparent hypothyroidism. So integrating number 2 and number 3 led me to hypothesize that remodeling of cardiac myosin via heat acclimation or climatization elicits beneficial adaptations in the heart. And to have a proof of this concept, we studied the myosin isoforms in the heat acclimated heart with respect to thyroxine level in the blood. And we measured both T4 and T3. And 3 weeks after the onset of acclimation, we saw a reduction of 50% of thyroxine level. It was only apparent reduction because it was only in the serum. But at that time, we saw a transition from fast myosin, which show in the control animals, which is in the control, was marked here by A, to a differential difference between the isoforms, and we saw the predominance of slow myosin, which is the V3. And after this 3 weeks, it maintained so throughout the entire acclimatization period. The match control continued to maintain the fast myosin. The transition from V1 to V3 myosin population is a tonic adaptive process to cope with high sustained demands for tension, like, for example, volume overload or impaired energy supply. A manifestation of the presence of the slow myosin predominance results in a greater pressure at lower oxygen consumption. And you can see it here on the left panel of the slide, that the black circles denote acclimated hearts of rats compared to the open circles, which are the control. In the axis, in the x-axis, we have the workload presented by rate pressure product, and you can see that the heat acclimated hearts maintained greater work at the same oxygen consumption of the controls which had the lower work efficiency. You can see it on the right panel in exercising animals, acclimated to heat compared to exercising alone. And again, if you can see in the lower panel of the left side, you can have, again, the black circles are the exercising. The x-axis is now oxygen consumption. And the exercising animals produced greater work at similar oxygen consumption as the control, which produced lower exercise, a lower ability to exercise. During acclimation, there is a significant expansion of plasma volume and in turn increased venous return. The heart, as opportunistic organ, responds by changing in elasticity. And you can see it here on the left channel by measurement of compliance. You can see that in the heat acclimated hearts, again, the black dot, there is increased large diastolic filling with no change in pressure, namely increased compliance. On the right side, you can see changes in stiffness, which is the change in pressure versus change in volume. And again, the dotted line denote decreased chamber stiffness in the heat acclimated animals. Heat acclimation, so in conclude, result in a change in the intrinsic properties of the acclimated heart exhibited by increased compliance, reduced chamber stiffness, and its application, which is used now by various clinicians in the investigation, is thermotherapy to reduce arterial stiffness in patients suffering from cardiovascular disease. The last issue I'd like to talk on is increased ischemic tolerance, and you can see it, and you can see it here by the development of ischemic contracture. This means the increase in diastolic pressure when during ischemia the heart ceases to work, and it occurs because of no available ATP, sufficient ATP to disconnect between the head of the myosin and the actin. On the right side, you can see sedentary acclimation, and when perfusion reduced by 50%, the acclimated animal, this is the yellow dot, show less ischemic contractures as the non-acclimated animal, but during exercise, both acclimation and exercise protect the heart from ischemic contracture. The other thing I would like to show is that on the left side, you have measurement using phosphorus NMR spectroscopy, and both the slices are of hearts, and you can see that only the C heart, which is on the left side, on the left, in the bottom panel in the left show infarct size, which is the white size, and all the rest, acclimated, sedentary, exercising, and sedentary and exercising with acclimation, are protected. One more important thing that because of lack of time, I don't like to discuss it, but acclimation make the heart, decreases calcium sensitivity, so the heart is protected from calcium overload. We see it both in the heart and in the brain in some other studies. And to summarize, I make a scheme that summarizes all what I told you so far. increase the venous return because changes of elasticity result in ability of the heart to receive more blood and increase stroke volume. There are biochemical adaptive modalities like changes in EC coupling, calcium protection, and together they increase the pressure production because of the presence of the slow myosin together with increased stroke volume result in increased cardiac output with greater efficiency. One last thing I'd like to mention, it's already all of these studies were conducted after 1998, but we know that within like epigenetic mechanisms leading to reprogramming of gene expression result in changes in mitochondrial function, cytoprotective protein, and some other features. But since we are not going to discuss cardiac physiology now, I'll stop here and leave the stage to my colleague.
So thank you, Michal. Thank you, Professor Horowitz. And based on everything we saw and the hard work done in the lab, we wanted to see if this is applicable to human beings, not only to animals. And we embarked on a clinical study trying to see if these mechanisms have any effect in humans. So in the background, the cardiopulmonary bypass that we use during coronary bypass operation, this mechanism during operation imposes myocardial ischemia followed by reperfusion. Both myocardial ischemia and reperfusion are known causes of diastolic dysfunction. So the distensibility of the heart is less good after coronary bypass than before. And in 1994, McKenney and colleagues from Boston University, which I personally know because I was at that time there as a fellow, although not participating in this intraoperative study that they published, they were using transesophageal echocardiography looking at the heart from the esophagus during operation, and they showed an increase in diastolic chamber stiffness immediately after coronary artery bypass surgery, reflecting worsening of diastolic function immediately after surgery. And the animal studies done by Professor Horowitz, which went so deeply into the mechanism, they indicated that heat acclimation has a protective effect against ischemic injury. So we hypothesized that heat acclimation may protect the heart from the deleterious effect of cardiopulmonary bypass on diastolic function. And we aimed to prospectively study the influence of heat acclimation with exercise together on diastolic dysfunction imposed by cardiopulmonary bypass in ischemic patients undergoing surgery. So we looked consecutively in patients that were scheduled to have coronary bypass because of severe ischemic heart disease. We excluded patients who had significant valvular disease, left ventricular hypertrophy, and reduced left ventricular function, because these are all factors known to have an effect and to influence diastolic function. And we came up with two groups of patients. The first was an 8-group patient, 8 patients that were the acclimatization group. How did we acclimatize them? We sent them to the Dead Sea seashore. We put them in caravans a little bit south from here near the hotel area. They were on the seashore for 2 to 3 weeks before operation, immediately before their operation. No air condition at that time. You can assume that it wasn't really very pleasant all day long. At night it was much better. And they also exercised twice daily, modest exercise. Remember, these were ischemic patients scheduled to have coronary bypass. So they couldn't do too much exercise, and it may have been even deleterious to them and Dr. Mara Shochina, who is here in the audience, who is an expert in rehabilitation and rehabilitation physiology, designed a specific protocol for each patient according to his level of exercise to be able to do this training for 3 weeks. To these 8 patients, we matched 8 other patients, and they were matched by age, sex, coronary disease severity, left ventricular diastolic dimension, left ventricular mass, and left ventricular systolic function. All factors that we thought may interfere in explaining the results. These patients also were scheduled at the same time period to undergo coronary artery bypass grafting, but they stayed home in their cities, up on the mountains, above the seashore, up to 700 meters, at altitude above up to 700 meters, so in colder areas. And when they came to the operation, our intraoperative protocol was based on transesophageal echocardiography images of the left ventricular short axis, and in the same time, simultaneously measurement of left atrial pressure. And the transesophageal echocardiography and left atrial pressure data were acquired both before and immediately after getting off the bypass machine with preload manipulation by fast volume loading. And the change in diastolic stiffness was assessed by comparing left ventricular and diastolic area at similar left atrial pressure pre and post cardiopulmonary bypass. And just for those who were not really very happy with the simplified physiology of the heart, I would just say that get the notion that for a given left atrial pressure, a smaller left ventricular area implies a leftward shift of the pressure-volume relationship, means a stiffer heart, less compliant, not good, while a larger left ventricular area suggests a rightward shift of the pressure-volume curve, and this means increased compliance, which we assume is good. These are the baseline and intraoperative variable of those 2 groups, 8 patients in each group. You can see they were well matched to start with. Age was about 66. Risk factors, hypertension, diabetes were the same. Chamber mass and chamber dimensions were the same. The number of vessels that had to be grafted during operation were the same, and so was the cardiopulmonary bypass time. The only thing that had a difference, a significant difference between the group was obviously the temperature they were exposed to before operation, in the period before operation. The mean temperature of the acclimatized group was about 23 degrees centigrade. The mean temperature of the non-acclimatized group was about 11, so on average there was a 12 degrees centigrade difference between those groups in the weeks before the operation. On the right side panel, you'll see a left ventricular pressure area relation in a representative non-acclimated patients. Before bypass, you see the black dots. This is the pressure volume loop that he had before bypass. Immediately after bypass, the white dots, there is a distinct leftward shift, which means stiffer ventricle, less compliant, not good. On the left panel, you see the pressure area relation in a representative acclimated patient who was here in the Dead Sea. You can see that there is virtually a superimposition of both graphs, meaning that before and after bypass, there were no decrease or no significant change in diastolic properties. And when we put all these patients together and look at the left ventricular pressure area relation before and after bypass, on the right side you see the non-acclimated group. You can see that before bypass with the black dot, there is a relationship that has a certain value, and this is parallel moving leftward immediately after bypass, so it's becoming stiffer, and this is highly significant. The acclimated patients did not have any change in their diastolic stiffness, and these graphs show that there has been a benefit by acclimating them. And therefore, we concluded that this was the first clinical study supporting the experimental evidence presented to you by Professor Horowitz for a protective effect of heat acclimatization with modest exercise training against diastolic dysfunction that was, in this case, the model was ischemia reperfusion during coronary bypass. And definitely, this is a very small study, but still very significant, we believe. And further clinical studies are justified to expand on this data, to evaluate the longevity of this effect. We don't know how long it lasts, and to see how relevant it is to the cure of the patient after coronary bypass. And I thank you for that, and this is Jerusalem at the Hadassah Medical Center, just in contrast to what we see outside. Thank you very much.